Hello friends, today I will talk about insulin. I will stress upon its structure and how does it affects the metabolism of the carbohydrates, that is glucose, fats and the proteins. First is the disclaimer. This video is made for educational purpose only and there is no endorsement for any brand or company. These are the four Nobel Prize winners who discovered the insulin, Mr. Banting, Mr. Best, MacLeod, James Collip, and the first Indian doctor who used insulin was Dr. J.P. Bose. Before discussing about the insulin, let's see the anatomy of the pancreas. As we know, the pancreas has mainly a digestive function, but it also secretes two very important hormones. They are insulin and glucagon. And these insulin and glucagon, they are important in the regulation of the glucose, lipids, proteins and the protein, lipids and the glucose metabolism. Pancreas has two types of cells, that acini and the islet of Langerhans. These are the acini cells and these are the islets of the Langerhans cells. Acini, they secrete digestive juices which go into the duodenum, and islets of the Langerhans, they secrete insulin and glucagon which directly go into the bloodstream. Pancreas has about 1 to 2 million islets of the Langerhans and they are arranged around the small capillaries in which they secrete the hormone. These islets have three types of cells, alpha, beta and delta, and beta cells, they comprise about 60% and they mainly secrete the insulin and amylin. Alpha cells, they constitute about 25% of the total and they secrete the glucagon, and delta cells, they account for 10% and they secrete the somatostatin. Coming to the structure of the insulin, as you know that insulin is a small protein or polypeptide. It has a molecular weight of 5800 daltons and it is composed of two amino acid chains, that is alpha and beta, which are connected by the disulfide bonds. Insulin is synthesized in the beta cells as a part of protein synthesis. To begin with, there is a translation of the insulin RNA by the ribosomes which are attached to the endoplasmic reticulum to form the pre-proinsulin. This initial pre-proinsulin has a molecular weight of 11,500 dalton, but it is then cleaved into endoplasmic reticulum to form a proinsulin with a molecular weight of about 9,000 daltons, and it consists of three chains of the peptide A, B and C. The proinsulin in the Golgi apparatus gets cleaved at a particular site to a metabolically and physiologically active part, that is the insulin, and it is composed of A and B chains connected by the disulfide linkage. And the C peptide, it is an inactive part. C means connecting peptide. Both insulin and C peptide, they are packed in the secretory granules and secreted in an equimolar amount. Insulin usually circulates in the blood in the unbound form. It has a very short half-life of about 6 minutes and it is mainly cleared from the circulation within 10 to 15 minutes. Insulinase is the enzyme which is primarily present in the liver and to a smaller extent in the kidneys and the muscles, which degrades the insulin in the tissues. So insulin, actual word is insula, means island. It is a peptide hormone. It contains about 51 amino acids. It has two chains, alpha and beta. This is alpha chain and this is beta chain. They are connected by the disulfide bond. Alpha chain has 21 amino acid and beta chain has 30 amino acid. Both chains are linked together by disulfide bonds. Just remember, for every single unit of insulin, equal amount of the C peptide is produced by the pancreatic beta cells. And importance of this is, if we want to know the amount of insulin which is produced by the beta cell in diabetic patient, just measure the C peptide level we will get the amount of the insulin. So C peptide level is basically the marker of endogenous insulin production. In type 1 diabetes mellitus, insulin deficiency is there, C peptide level will be undetectable or they will be very low except during the honeymoon period. And in case of type 2 diabetes mellitus, there is increased insulin resistance, so C peptide level will be either normal or high. So coming to the mechanism of insulin secretion, as we know that insulin is secreted in the pancreas in response to the blood glucose levels. The beta cells, they have a very large number of glucose transporters that permit the rate of glucose influx. These beta cells have uh, glucose transporters. These are the glucose transporters that permit the rate of glucose influx that is proportional to the blood concentration in the physiological range. Once inside the cells, once this glucose enters the cells, it is converted by the glucokinase into glucose 6 phosphate, and which further undergo oxidation to form the ATP, that is adenosine triphosphate. This ATP inhibits the ATP sensitive potassium channel. These potassium channels, ATP sensitive potassium channels, are inhibited when the glucose enters the cells. Glucokinase is activated. It converts the glucose into glucose 6 phosphate, and this glucose 6 phosphate is subsequently oxidized to adenosine triphosphate, that is ATP, which inhibits the ATP sensitive potassium channels of the cell, and the closure of these potassium channel depolarizes the cell membrane and thus leading to the opening of the calcium channels, voltage gated calcium channel, which are sensitive to changes in the membrane voltage. This effect produces an influx of calcium. So there is influx of the calcium into the cells, and this influx of the calcium stimulates the fusion of the docked insulin containing vesicle within the cell membrane, and which leads to a secretion of the insulin into the extracellular fluid by the exocytosis. So whenever glucose enters the cell, it gets converted into glucose 6 phosphate by glucokinase. Glucose 6 phosphate gets oxidized into ATP. ATP causes potassium channel closing. When these potassium channel are closed, there is opening of the calcium channel. There is influx of the calcium, and influx of the calcium leads to the secretion of the insulin from the cell into the extracellular fluid. There are certain hormones such as glucagon, glucose dependent insulinotropic peptide like GIP, and acetylcholine. They also increases the intracellular calcium level, though through other signaling pathway, and they can enhance the effect of glucose, but they do not have a very major effect on insulin secretion in the absence of the glucose. There are other hormones also like somatostatin, norepinephrine. By activating the alpha adrenergic receptors, they inhibit the exocytosis of the insulin. Sulfonylurease drugs, they stimulate the insulin secretion by binding to ATP sensitive potassium channel and blocking their activity. This mechanism results in depolarization effect that triggers the insulin secretion and making these drugs useful in stimulating insulin secretion in the patient with type 2 diabetes. Sulfonylureas, basically these are they enhance the insulin secretion from the pancreas. So if we see that how the insulin secretion is controlled by the blood glucose, whenever there is increased blood glucose, it stimulates the insulin secretion. At normal fasting level of blood glucose like 80 to 90 milligram per deciliter, the rate of insulin secretion is only about 25 nanogram per minute per kg of the body weight. This has a very has only slight physiological activity. If the blood glucose concentration is suddenly increased to level of 2 to 3 times normal and is kept at this higher level, then the insulin secretion increases markedly into two stages. So initially, when there is normal fasting blood glucose level, that is between 80 to 90, there is slight increase in insulin secretion from 25 in the order of 25 nanogram per minute per kg body weight. But when their glucose concentration is very high, especially when we take the meal, that its blood glucose level increases 2-3 times, then at that time insulin is secreted in two stages. And the how is secreted in the two stages? The concentration of the insulin in the plasma increases almost tenfold within 3 to 5 minutes. So this we can see within 5 to 10 minutes there is increase in the plasma insulin level in the after the acute elevation of the blood glucose. This increase result from the immediate dumping of the this increase is because of the immediate dumping of the preformed insulin from the beta cells of the islets of the Langerhans. However, the initial high rate of secretion is not maintained. Instead, the insulin concentration decreases about halfway back towards normal in another 5 to 10 minutes. So, whenever there is, when we take food, there is tenfold increase in uh, secretion of the insulin in the initial 3 to 5 minutes. Then insulin concentration again forms, and this insulin is released because of the initial preformed vesicles which contain the insulin. They release those insulin. So and this returns back to normal in another 5 to 10 minutes. At about 15 minutes, insulin secretion rises a second time and reaches a new plateau in 2 to 3 hours. Then thereafter, again the insulin secretion increases and it reaches a plateau in 2 to 3 hours. And this is because of additional release of preformed insulin and from the activation of the enzyme that synthesize and release new insulin. And this peak is because of new insulin release and also from the preformed insulin which was already there in the vesicles. So the insulin rises very rapidly and it reaches a peak of at least about 10 to 25 times the basal levels, 10 to 25 from the basal levels, whenever there is blood glucose is in the range of 400 to 600 milligram. So insulin is secreted in two stages. First stage is first rise within 2 to 3 minutes or 3 to 5 minutes, tenfold rise. It is because of the preformed insulin. And second rise is because of the both preformed insulin and new insulin secretion. So this is very important to learn. There are other factors also which they also secrete which help in secretion of the insulin. There are certain amino acid, especially arginine and the lysine. Not all amino acids cause insulin secretion. These two amino acids like arginine and lysine, they also have insulin secretion effects like glucose, but they act only in the presence of insulin. Whenever these amino acids are administered in the absence of the rise in blood glucose, they cause only a small increase in insulin secretion. However, when they are administered at the same time that the blood glucose concentration is elevated, the glucose induced secretion is insulin may be as much as doubled. So when they are administered alone, then the rise in insulin is not up to the mark. But when they are administered along with the glucose or whenever the blood glucose level is already elevated, then the insulin secretion gets doubled. These amino acids strongly potentiate the glucose stimulus for insulin secretion. This stimulation amino acid insulin secretion is important because the insulin in turn promotes the transport of amino acids into the tissues as well as intracellular formation of proteins, that is protein synthesis. That is, insulin is important for proper utilization of the excess amino acid in the same way as it is important for the utilization of the carbohydrate. There are certain gastrointestinal hormones like gastrin, secretin, cholecystokinin, GLP-1 glucagon, that is glucagon like peptide 1, and glucose dependent insulinotropic peptide. They can cause moderate increase in insulin secretion. The two important hormones like GLP-1 and GIP, they are potent hormones. They are also called incretin. They cause increase in insulin release from the pancreatic beta cell in response to increase in the plasma glucose. So they also inhibit the glucagon secretion. So GLP-1 and GIP, they cause increase in insulin secretion when uh, patient eats meal or patient takes glucose. They also inhibit the glucose secretion. So these GLP-1 and GIP are nowadays they are used in control of blood sugar. Their analogs are available. There are other hormones and autonomic nervous systems like glucagon, other hormones like glucagon, growth hormone, cortisol, and to a lesser extent progesterone and estrogen. They also uh, cause increase in insulin secretions. The importance of the stimulatory effect of these hormones is prolonged when they are when they are given for a prolonged period of time, or they are present in a prolonged period of time, then they cause increased secretion of the insulin from the body, and they may lead to exhaustion of the body beta cells in the pancreas, and thereby they may increase the risk of developing diabetes mellitus. Diabetes is very particularly common in patients who have acromegaly or gigantism, or those patients who have adrenal tumor which secretes excess of glucocorticoids. The pancreatic beta cells or islet cells are richly innervated by the sympathetic and the parasympathetic nervous system also. Whenever there is stimulation of the parasympathetic nervous system, pancreas this leads to increased insulin secretion during the hyperglycemic conditions, whereas when sympathetic nervous sympathetic nerves, they stimulate, uh, they cause increased glucagon secretion, and they decrease the insulin secretion during the period of hypoglycemia. So the glucose concentration are believed to be detected by the specialized neuron in the hypothalamus in the brainstem, as well as by the glucose sensing cells in the peripheral locations such as liver. So coming to the physiological actions of the liver, as we know that it is the main anabolic hormone in the body. It regulates the metabolism of the carbohydrates, fats, and the proteins. So how does it affects the metabolism of the glucose or the carbohydrates? Insulin, it increases the uptake of glucose from the blood by the liver cells, thereby increasing the activity by increasing the activity of the enzyme glucokinase, which causes initial phosphorylation of the glucose after it diffuses into the liver cells. Once the glucose enters the liver cells, it gets phosphorylated, and this glucose is temporarily trapped inside the liver cells. So it increases the uptake of the glucose in the liver by the liver cells by activating the enzyme glucokinase. It also increases the activity of the enzyme which promote the glycogen synthesis. So it promotes the activity of which enzyme? That is glycogen synthesis. So increases the glycogen synthesis also. It also inactivates the enzyme liver phosphorylase. This phosphorylase enzyme causes a splitting of the glycogen into the glucose. So when this enzyme is inactivated by the insulin, there will be no breakdown of the glycogen, and there will be no release of the glucose from the uh, liver cells by the glycogen. So net effect of all these action is there is increased amount of the glycogen synthesis in the liver by the insulin. When the glucose is released from the glucose is also released from the liver between the meals. When the blood glucose level in the body falls to a low level, then there is decreased blood glucose concentration. It causes the pancreas to decrease its insulin secretion also. The lack of insulin then reverses all these four above mentioned effects, and which further stops synthesis of the glycogen in the liver, and thus preventing the further uptake of the glucose by the liver in the blood. The lack of insulin, along with the increased release of the glucagon, activates the enzyme phosphorylase, which causes the splitting of the glycogen into the glucose phosphate. Means whenever there is insulin deficiency, the enzyme phosphorylase will get activated, and it will split the glycogen to the glucose. The enzyme glucose phosphatase, which has been inhibited by the insulin, now becomes activated because of insulin deficiencies, and the phosphate radicals, they split away the glucose and allowing the free glucose which diffuse back to blood. Insulin also promotes the conversion of the excess glucose which we take into fatty acids and inhibits the gluconeogenesis in the liver. So when the quantity of the glucose entering the liver cell is more then that can be stored as a glycogen or can be used for local hepatocyte metabolism. Means when the quantity of the glucose entering the liver cell is more than can be stored as a glycogen or can be used for local hepatocyte metabolism, then insulin promotes the conversion of all these excess glucose into the fatty acid, and these fatty acids are subsequently packaged as a triglyceride into very low density lipoprotein, and which are transported in the form by the way of the blood to the adipose tissue, and they are deposited there as a fat. Insulin inhibits the gluconeogenesis mainly by decreasing the quantities and the activities of the liver enzyme required for the gluconeogenesis. So whenever there is lack of insulin, what happens to the brain? The brain is quite different from the other tissues in using the glucose. Whenever there is insulin deficiency, or whenever there is decreased blood glucose levels in the body, then initially the brain uses the glucose only for its energy purpose, but if it is not available then it can use fats also. So the brain is quite different from most of the other tissues of the body, because insulin has a very little effect on the uptake or use of glucose by the brain. So the most of the brain cells, they are permeable to glucose and they use glucose without any intermediation of the insulin. The brain cells normally use only glucose for energy and use other substrate for substrates such as fats only with the difficulty. Therefore, it is essential that blood glucose levels always be maintained above the critical level. When the blood glucose level falls too low, that is in the range of 20 to 50 milligram per ml per 100 ml, the symptoms of hypoglycemia like nervous irritability leading to fainting, seizure, and even coma can occur. Effects of insulin on fat metabolism. It's not seen in a short term, but we usually see how does the insulin affects in long run. It has been seen that long term insulin deficiency causes atherosclerosis, which leads to heart attack, cerebral strokes, or other vascular accidents. So insulin actually promotes fat synthesis and its storage. Firstly, the insulin increases the utilization of glucose by the most of the body's tissue, which automatically decreases the utilization of the fat. So initial uh, insulin has a fat sparing effect. However, insulin also promotes the fatty acid synthesis, especially when more carbohydrates are ingested, thus providing them as a substrate for fat synthesis mostly in the liver cells. And these fatty acids are then transported from the liver by the way of the blood lipoprotein to the adipose cells to be stored. Uh, how does it affect? Increase insulin. Insulin increases the transport of glucose into the liver cells, and this when this glucose reaches the liver cell, it gets split into the pyruvate in the glycolytic pathway, and this pyruvate subsequently gets converted into acetyl CoA enzyme. This acetyl CoA then further gets converted into malonyl CoA, and this subsequently leads to fatty acid synthesis. So overall effect of insulin is in the fatty acid synthesis and the storage of the fat as a triglyceride. So insulin has two essential effects in the fat storage as a adipose tissue. It inhibits the lipase enzymes. This lipase causes the hydrolysis of the triglyceride already stored in the fat cells. So uh, as it inhibits the lipase, so the fatty acids will not be released from the adipose tissue once they are stored, and if the insulin is present in a sufficient amount. Insulin also promotes the glucose transport through the cell membrane of the fat cells in the same ways as it promotes the glucose transport into the muscle cells. and some of this glucose is utilized in synthesis of the fatty acid, but more importantly it also forms a s- and forms large quantity of alpha glycerol subphosphate, and this alpha glycerol phosphate supplies the glycerol that combines with fatty acid to form triglyceride, which are the storage form of fat in the adipose cells. Therefore, when the insulin is not available, even the storage of large amount of fatty acid transported from the liver in lipoprotein is almost blocked. So, what will happen if there is insulin deficiency? In the state of insulin deficiency, the fat will break down and its use for providing energy are generally enhanced in the absence of insulin, and this enhancements occurs even normally between the meals when secretion of the insulin is minimal, but it becomes extreme in person with diabetes mellitus, and the secretion of the insulin is almost when the secretion of the insulin is almost zero. So insulin deficiency causes lipolysis of the storage fat and release of the free fatty acids. In the absence of insulin, all the effects of insulin which I described earlier, like storage of fat, they are all reversed, and the most important effect is in the lipase. Lipase gets activated, and this activation causes the hydrolysis of the stored triglyceride, releasing large quantity of fatty acid and glycerol into the circulating blood, and these plasma concentration of the free fatty acid begin to rise within minutes in state of insulin deficiency, and they become free fatty acid, they become free main energy substrate for all tissue except the brain. So this we can see in a case if we remove the pancreas, how does the free fatty acid level, how does the blood glucose level rises and acid ketone body level rises. The it has been seen that in a prolonged state of insulin deficiency as seen in diabetes patients, especially type 1 diabetes patient, there is increased risk of development of atherosclerosis. So excess in patients of insulin deficiency, it has been seen if there is excess release of the fatty acids, there is very high chances of the ketone body formation leading to ketoacidosis. Insulin deficiency actually causes excess amount of acetic acid to be formed in the liver, because what happens in the absence of insulin, there is excess presence of fatty acid in the liver, and the carnitine transport mechanism gets activated in the mitochondria, and in mitochondria the beta oxidation of the fatty acids occurs, and this leads to extreme increase in amount of acetyl CoA coenzyme A, and the large part of this excess acetyl coenzyme A then condensed to form acetic acid, which in turn is released into the circulation, and most of this acetoacidic acid passes to the peripheral cell where it gets again converted into acetyl coenzyme A, and it is used for energy in the usual manner. But at the same time, because of absence of insulin, there also occur depression of the utilization of these acetoacidic acid in the peripheral tissue, and they get starts accumulating in the blood or the tissue, and the concentration of these acetoacetic acid rises during the days after cessation of the insulin secretion, and sometimes the concentration may go beyond 10 milliequivalent per liter or more, and which in severe state of body fluid, which leads to severe state of acidosis, and some of these acetoacetic acid also gets converted into beta hydroxybutyric acid and the acetone, and these are basically ketone bodies, and when they are present in the large quantities they leads to a state of ketoacidosis, and these ketoacidosis uh, when developed in a patient, they may lead patient to go into coma, and eventually the patient have may face the mortality. Let us discuss about the insulin, how does it affects the protein metabolism? Insulin stimulates the transports of many of the amino acid in the cells, and especially valine, leucine, isoleucine, tyrosine, and phenylalanine. But the transport of insulin or synthesis of the proteins uh, usually occurs uh, when insulin acts synergistically with the growth hormone. Insulin basically increases the translation of the messenger RNA, and it leads to formation of the new proteins, and in some unexplained way insulin turns on the ribosomal machinery, and in the absence of insulin the ribosome simply stop working, and almost if the insulin is deficient there may occur decreased synthesis of the protein. So over a longer period of time insulin increases the rate of transcription of the selected DNA and leads to more protein synthesis. In a nutshell, we can say that insulin promotes the formation of proteins and prevents the catabolism, that is the degradation of the proteins. And in cases of insulin deficiency, we have seen the protein depletion and increase in the level of the plasma amino acid. Virtually all protein storage comes to halt whenever there is insulin deficiency, and this leads to the catabolism of the protein, and protein synthesis stops. Large amount of the amino acids are uh, they go into the circulation, and these amino acid concentration in the plasma, when this rises and most of the excess these amino acids are they are used directly for energy, or they act as a substrate for gluconeogenesis, and this degradation of amino acids leads to enhanced urea excretion in the urine, and the resulting protein wasting is one of the most serious effect of the severe diabetes mellitus. So the diabetic patient may feel very extreme weakness, and they may have deranged uh, function of the other organs also. So insulin and growth hormones, they interact synergistically to promote the growth. Because insulin is required for the synthesis of protein, it is an essential it is as essential as growth hormone for the growth of an animal. It has been seen that the two hormones, they function synergistically to promote the growth, with each performing a specific function separate from that of other. And it has been seen that small part of necessary for both hormones result from the fact that each hormone promotes cellular uptake of different selections of amino acid, and all of which are required for the growth required if the growth is to be achieved. So in a nutshell we can say, for growth, both hormone they act, insulin and the growth hormone, both hormone act synergistically, and without one without the one hormone there is complete growth may not be achieved. So their action is very synergistic. The role of insulin and other hormone in switching between the carbohydrate and the lipid metabolism. As we know that when glucose concentration is low, insulin secretion is suppressed, and fat is used almost exclusively for the energy expenditure, except in the brain. When the glucose concentration is high, insulin secretion is stimulated and carbohydrate is used instead of fat. The excess of the blood glucose is stored in the liver as a glycogen, liver fat, or the muscle glycogen. One of the most important functional role of insulin in the body is to control which of these two foods will be utilized by the cell for energy from moment to moment. Four hormones, they have an important role to play in this switching mechanism. Like growth hormone, it is secreted by the anterior pituitary gland, cortisol secreted from the adrenal cortex, epinephrine secreted from the adrenal medulla, and glucagon from the alpha cells of the islets of the Langerhans and the pancreas. Both hormones, like growth hormone and the cortisol, they are secreted in response to hypoglycemia, and both inhibit the cellular utilization of the glucose while promoting the fat utilization. However, the effect of both these hormones develops slowly and usually require many hours for proximal expression. sorry, for the maximal expression. Epinephrine instead, it is important in increasing the plasma glucose concentration during the periods of the stress when there is sympathetic nervous system gets excited. However, the epinephrine act differently from the other hormone in that it increases the plasma fatty acid concentration at the same time. So this epinephrine increases the fat concentration and other glucose concentration also. The reason for this effect is epinephrine has a potent effect on causing glycogenolysis in the liver, thus releasing the large quantity of glucose in the bloodstream within minutes. And it has also a direct lipolytic effect on the adipose cells, and it activates adipose tissue lipase, thus generating blood large amount of fatty acids in the blood circulation. So if we see the quantitative effect of the epinephrine, the enhancement of the fatty acid is far greater than the enhancement of the blood glucose. So the release of the fatty acid is more as compared to the glucose when the epinephrine acts in a state of uh, sympathetic overactivity. So it has been seen that epinephrine especially enhances the utilization of fat in stressful states such as exercise, circulatory shock, or anxiety. So this was all about insulin. I tried to discuss about the structure of the insulin, its role in the metabolism of the carbohydrates, fats, and protein. Overall, we have seen that insulin is a basically anabolic hormone. It increases the uptake of glucose in the liver, skeletal muscles, brains. Though it has a very little role in the brain, it promotes the fatty acid synthesis, it promotes the protein synthesis, and in the absence of insulin, we have seen there occurs lipolysis, increased catabolic state in the body, and it plays a very important role. If there is deficiency of insulin, then the patient may have diabetes mellitus, which I will be discussing in my next discussions. The reference for this talk were taken from the Guyton Hall textbook of medical physiology, 13th edition. Thank you very much.